Oops. Hello, everyone. Thank you for joining today's HUPO Education and Training Committee webinar series, Use of Proteomics in Clinical Investigation. The Education and Training Committee develops initiatives, including workshops, courses, and webinars, promoting global learning and collaboration. Today's webinar is the fourth in this series. We encourage you to explore past recordings on our YouTube videos and also stay updated at the HUPO website. This editorial was published this year, summarizing our education and committee's past webinars. We look forward to do the same in the future. Now, I'm pleased to turn it over to my co-chair, Dr. Yansen Liu, professor at Yale University. And I am from Baylor College of Medicine, Houston, Texas. So Dr. Liu will introduce our distinguished speaker for today. Yansen?
Okay, great. Thank you, Shisha. So today we are very honored to have Professor Joshua LaBaer with us today as our speaker. So just a quick introduction about Josh. Joshua LaBaer is an internationally recognized leader in the personalized diagnostics and proteomics. He holds an MD and PhD from UCSF and now serves as the executive director of the Biodesign Institute at Arizona State University. As a pioneer in clinical proteomics, Josh invented the nucleic acid programmable protein array and has advanced the biomarker discovery for diseases such as cancer, diabetes, and COVID-19. Previously, Josh established the first U.S. clinical lab to offer public testing for COVID-19 and now also co-lead the NIH SeroNet effort to monitor the immune responses. He was elected as a fellow of the National Academy of Inventors, received the HUPO Translational Proteomic Award as early as 2016, and served as the past president of US HUPO, and also authored over 250 scientific publications. Okay, so I will stop here. Just want to remind everyone that please feel free to use the Q&A in the Zoom to ask your questions. And Josh, please take it away.
Awesome. Thank you so much for that kind introduction. Let's see if I can get my screen to work here.
Yes, it's sharing.
Yeah, I need to switch it though, I think.
Mm-hmm.
All right. Are we good here?
Yes.
Okay,
Yes.
great.
Perfect.
Well, thanks so much for inviting me. I'm really excited to tell you about some of the newer things going on in our group. I'm going to just begin by motivating this by kind of telling you why we do what we do. This is a plot of survival of breast cancer depending on the stage of diagnosis. So across the bottom of this screen, you can see different stages of when cancer is diagnosed. And clearly, women who are diagnosed with breast cancer in an earlier stage have a very good outcome. 100% of them have a survival rate in 5 years compared to later stage diagnosis. So one of our big goals is to be able to diagnose cancer as early as possible. And one approach that we're particularly interested in to look for markers, blood-based biomarkers for cancer, is to look at what are called autoantibodies. So normal cells, either when they die, they release proteins into the serum, or they secrete proteins. And then the immune system typically ignores proteins that come from normal cells because of immune tolerance, which means that our bodies should not be making antibodies against self-proteins. But in the case of cancer, cancer cells sometimes just secrete abnormal proteins or abnormal amounts of proteins that act as neoantigens, and they can induce antibodies to these antigens, specifically in the context of cancer. And these are called autoantibodies. And they have advantages as biomarkers because even if the amount of the protein here was not very much, there's often much more antibody available to detect. So the antibodies effectively amplify the signal. And even if these proteins disappear, the antibodies are still around. And so they become useful markers for the presence of cancer. The question is, how do we know which proteins trigger an immune response in cancer? And the classic way to measure immune responses is the ELISA assay, where you put proteins in the wells of a 96-well dish. You add antibodies to each well, and if you get a signal like this one here, that person has a strong response to that protein, and this person has no response to that protein. The challenge, of course, is that it takes a lot of protein to coat these wells, and you're typically looking at them only one at a time, and so that really slows the process down. So we and others have considered this approach of looking at what are called protein microarrays to track immune responses. The idea here is to have a slide or another matrix that has thousands of different spots on it, each one a different protein. We add serum to that array, and then anywhere something lights up, that is an antibody response to that protein. And what you can then do is you can look at a bunch of arrays for serums for patients who have cancer and compare them to arrays for patients who do not, and look for those signals that are specific to the cancer patients. So we set out to build protein microarrays a number of years ago. This is work that Neera Ramachandran and Jeannie Hainsworth really pioneered, along with several other folks who also contributed to that early work. This was the first published array. That was not from our laboratory. This is Gavin Macbeth when he was in Stu Schreiber's laboratory, also at Harvard when we were at Harvard. This array had 10,000 features on it. And here's the spot that Gavin was looking for. The challenge, though, was that his approach was to purify the proteins and spot them on the array. And consequently, this 10,000 protein spot array actually had only 2 proteins on it, 9,999 spots of one protein and 1 spot of the other protein. And that's because to do this with 10,000 different proteins, Gavin would have had to purify 10,000 different proteins and then spot them. Now, there are people who do that. Hung Ju at Johns Hopkins does a fine job of high throughput protein purification. Here's an example of their sorts of arrays. But there are challenges with this approach. The yields of protein when you purify them, particularly in high throughput, are highly variable, which means that will be reflected as variable spots on the arrays. And the shelf life of proteins, once you print them, varies. And so those proteins may not stay well folded after all that processing. So the approach that our group came up with years ago was, instead of printing the proteins, we print the genes, the open reading frames of the proteins on the array. So the array effectively, when we print it, is a DNA array. And we store that array until the day of experiment, when we add a cell-free extract that does in situ transcription and translation. It produces the protein. The protein is captured to the spot where it's made. And now we're presenting fresh protein on the array. And we don't have to worry so much about all the challenges. This is just an example of what that looks like. Here's the array prior to expression, so just staining it for DNA, because it begins as a DNA array. We add cell-free expression system, and now you see the proteins are being made. And what we've found over the years is that it's works for pretty much all proteins, membrane proteins, transcription factors, kinases, so soluble proteins, large, medium, and small proteins. We don't have to purify the proteins, and we get very consistent protein levels. All of our proteins are within a single log. We're using human ribosomes, so we're getting natural folding. And because they're full-length proteins, of course, we're getting both linear and conformational epitopes. So, we've liked this approach, and we've been using it for now many years. This is just to illustrate that you don't necessarily have to make full proteins if you want to map something. So here, Rodrigo Barderas was trying to map the epitope for an anti-p53 antibody. It was a monoclonal. He made a series of N-terminal deletions and a series of C-terminal deletions. And you can see when you add the antibody, it binds to all these N-terminal deletions, and then it stops binding. And from the C-terminal end, it binds to these and then not these. And so he could, look for the copy point where it lost, and it was around here. And he also built a series of tiling fragments that walked across the protein and showed that the antibody only bound to the fragment that correlated with this spot. So you can do parts of proteins if you need to. So how does this play in terms of biomarker screening? Well, here's the general approach. You're going to take these arrays that have thousands of proteins on it. I should mention that at this point, our lab now has over 18,000 human full-length genes cloned. So we cover most of the human proteome. It's really just the really large genes that we don't have anymore, but almost all genes we have. So we can really screen pretty much anything. Plus, we have tens of thousands of genes from other organisms. Print those on arrays, and then take serum from either patients or controls, and look for signals. And there are really several types of signals you might see. You're going to see a few of these blue spots. They're present in everybody. And we just had a paper a few years ago where we found a number of features that many people tend to have antibodies to. We're not entirely sure why, but those will show up frequently. You'll see a bunch of this individual variation. A lot of people will have antibodies at things that they've just developed over the years. And that really probably just represents their various exposures, possibly some mimicry. We're not entirely sure what, but there's definitely a lot of individual variation. But what's in red is what we're interested in, features that show up only in cancer patients and not in healthy controls. And these are the ones that we want to explore as potential biomarkers. This is what it looks like in real life. So this is an array stained for DNA, an array stained for protein after we've made the protein, and then patient and control sera. And we're looking at these features here that are lighting up as evidence that these might be antibodies that point to cancer. Now, you only know that after you do a large population study and then do a validation study. And I'll show you that in a moment. So this was one of the first, or was the first really, biomarker study we did in cancer. It started with 53 cases of breast cancer and 53 controls. At that time, we only had 5,000 human antigens. Now, as I mentioned, we have over 18,000. And we screened them to look for any features that were effectively uninformative. So that is to say, anything that gave no signal on either cases or controls, or which was the same in cases and controls. And we eliminated those. And that got us down to 760 antigens, which we could then print in duplicate. And then we compared 51 cases of breast cancer to 39 controls. In this case, our controls were not healthy patients. They were women who had non-cancerous breast disease. So things like cysts or abnormal findings, those were, we didn't look at those, but nonetheless, because we want to be able to look at cancer versus, benign disease. From there, we identified 120, almost 120 antigens that could distinguish between cancer and benign disease. And we did a third phase of validation. This time it was blinded, so we didn't know which cases were which. We compared 50 cases to around 40 controls, and we identified a 28 antigen panel that even after two rounds of validation could distinguish cancer from benign disease. And these are the antigens that we identified. At 98% specificity, this is what the sensitivity was. Typically, these autoantibodies have sensitivities between 15 and 40%. And that's pretty much what you see here. So, things like this one is kind of on the weaker end, or this one on the weaker end, and then others like this one, much stronger sensitivity. Since then, these markers were licensed out to a company. Initially, it went to this company, Videssa, which did a prospective clinical trial combining our antibodies with some protein biomarkers for cancer, and got an outstanding negative predictive value, around 97, 98%, both in young and older women. And they were using this test in combination with mammography to rule out women who likely didn't have cancer. As you know, mammography has about an 80% false positive rate. So 4 out of 5 findings on a mammography turn out not to be cancer. So this was a way of helping avoid biopsies in women who very likely did not have cancer. Since then, Provista has been shuttered. They had other business-related issues. And so the marker set has been sold to Totus Medical. But this is their product that they were looking at called Videssa Breast. So I've been showing you arrays from our earliest period of arrays. Those arrays typically had around 2,000 proteins per slide. What I want to mention now is that the team has developed a much higher density array. This is what we call our high density arrays. We do this by using the same technology to make computer chips. We use photolithography to etch nanowells in silicon that look like this. And then we use a piezoelectric printer to spit DNA into these wells, and then a specialized sealer to seal the wells one from the other so there's no cross-contamination. And so this allows us to get, at present, up to 20,000 features per slide. So really a 10-fold increase in throughput by using this generalized approach. It looks something like this. So you see very high density features on the array. But if you look at individual responses, they are quite discrete, and you don't see any cross-contamination because of the sealing. So we still use, this is the technology we use now. These really high density arrays really make screening much faster. So I'm going to stop here and say that, so what I've told you so far is technology that we've been using now for many, many years, and we've probably got dozens of publications that have used that protein array technology. Where that protein array technology really excels is if you have a large number of proteins that could be important and you don't know which ones to pick. And so, as I said, we can now screen 18,000 proteins from the human and tens of thousands of proteins from other organisms. So it's when you have a large number of proteins to test. The challenge with that approach is that it is expensive to look at 18,000 different proteins. You can imagine anytime you do anything 18,000 times, even if it were 10 cents a piece, that starts to add up to real money. And so when you're doing a screen at the beginning and you want to determine what are the best ones to focus on, the arrays are perfect. But as quickly as possible, once you've determined that you've whittled your numbers from 18,000 down to 100 or 200 different things, you don't want to keep doing the arrays. You want to find a simpler, less expensive approach. And so we were thinking a lot about how do we do high throughput screening in large populations when you didn't need to look at 18,000, maybe you wanted to look at 100 or 200. And that's the work. This is work started by Femina Ralph, really perfected by Lusheng Song, and really supported a lot by Jay Park and Ji Cho. So as I mentioned, the things we were looking for, the criteria we wanted to be able to look at thousands of samples. So not tens of samples, not hundreds of samples, but literally thousands of samples. We wanted to be able to still look at many antigens, but just hundreds, not thousands of them. Looking at things like vaccine versus natural infection, looking at things like different viruses. We wanted to expand the dynamic range. So on protein arrays, just about everybody uses fluorescence. Fluorescence has kind of a workable linear range of maybe 1.5 logs. We wanted to get that up to several logs. And then we wanted to get in solution binding. And we did that because the arrays fix the protein on the surface of a matrix. That limits the degrees of freedom of movement of that protein, and it makes it a little bit harder for things to bind to those proteins. So if you can get the protein fully in solution, the way molecules really are in life, you would get more natural binding. We wanted to, of course, continue to preserve the natural folding that we had with the protein arrays. So we wanted to continue using human ribosomes where possible, and eventually wanted this to be compatible with doing clinical studies. So the approach we came up with was to make our proteins and then barcode them with DNA barcodes. So this represents kind of a collection of different proteins. Each one has a DNA barcode. Then we use the antibodies from the patient to pull away anything that the patient recognizes. That separates the proteins that are recognized with patient serum from those that are not. And then all we have to do is read the barcode. So this becomes much easier than having to do the whole fluorescence thing and the protein array thing. So this is what it looks like in practice. We build a library of barcoded proteins. We incubate that mix of proteins with barcodes with patient sample. Typically, we really only use a microliter of serum. We typically pipette 5 microliters of diluted serum just because it's more accurate, but you don't need much serum. One to a few microliters is all that's needed. Bind the proteins to the antibodies, and then collect the antibody-bound proteins, and then just do a quick PCR to amplify the barcode. At the same time that you PCR the barcode, you add a sample barcode that is essentially an index that identifies anything in that sample. And that way you can distinguish bound proteins from this sample from any other sample. And so we do that for many samples. We pool them, we basically run them through a DNA sequencer, and then we can deconvolute that data to tell us who bound what proteins. What we've learned in the practice of this is that we can typically run anywhere between 2,000 and 10,000 samples per assay. We have a multiplexity of anywhere from 10 to 1,000 antigens per assay. Our dynamic range is around 3 to 6 logs. And because it's, of course, DNA sequencing, we believe it's compatible with the clinic. So I'll show you a little bit about what that looks like. And of course, both the in-solution binding and there's some agreement with commercial assays. So the first thing to point out about this approach is that it's highly reproducible. Here, what we're comparing are one run to a separate run on the same sample, so run on separate days. And we're looking at antibodies to a particular protein. This is the spike protein from SARS-CoV-2. This is a nucleocapsid protein from SARS-CoV-2. This is the Epstein-Barr virus, EBNA1 antigen, and this is a seasonal coronavirus. This is a bunch of different people, something like 180 different people. And you can see that their reading on one day is virtually identical to their reading on another day. So this is comparing one day to another day, and they line up beautifully on this axis. There's a couple of outliers here, but by and large, it's quite reproducible. Here, what we've done is take the same sample, measure it against many different antigens, so 147 different proteins, 10 repeats on each day, and compare them on 2 different days. And again, you can see that things line up beautifully. Error bars are quite tight, and you can really use this assay quite reliably over and over again. We then compared the platform to clinical assays to see if it agreed well with those. The first experiment we did was to look at samples collected by the Mayo Clinic from patients who had COVID early in the COVID pandemic, and compare them to samples from people taken pre-2019, so they never were exposed to COVID. And we got a very good distinction between the two. We then compared it to a FDA-cleared assay, the DX Labs assay. What you can see is in red are the DX Labs positive samples and the DX Labs negative samples versus our MISPA positive samples and our negative samples. MISPA stands for a multiplexed in situ protein array. And you can see that by and large, we do very well. We seem to pick up a number of things that the DX Labs called negative that we see strong responses to. They did pick up a few things that we called negative that we didn't see a strong response to. We also looked at using 5-parameter logistics, what the range of the assay was. It's around a 5 log range, but the linear range is really around 2 to 3 logs here. You can see that on this curve, and you can also get a sense of the reproducibility there. Here's another comparison of different populations between our platform and, again, another FDA-cleared commercial assay, looking at individuals with responses to the spike protein who either were not vaccinated nor had infection, were vaccinated but never had an infection, were not vaccinated but did have an infection, had both an infection and vaccination, or people who had multiple infections. And just looking from left to right, you can see that the two assays are very, very similar in terms of their output and who they identified. So very good agreement of this platform. The difference is that this clinical assay measures a handful of proteins, whereas this assay can measure 200 proteins at the same time. We also compared the platform to ELISA's. This is basically a classic ELISA for a different set of proteins. These are human papillomavirus proteins. And what we observed is on the x-axis is the ELISA, so you can see a 4-log range there, and our multiplexed assay on the y-axis. And what we observed was there are times when, this is a good example, the ELISA doesn't detect responses in all of these proteins, but we clearly saw antibodies to these proteins. So this platform seems to be more sensitive than ELISA in terms of detecting some of these responses. Here's, again, a number of responses that ELISA called negative that we clearly saw responses to. So it's rarely the other way around, that the ELISA is more sensitive than the MISPA assay. So we think that's probably due to the in-solution binding, the more accessibility of the antigen to the antibodies. So our first large-scale study was a volunteer study at Arizona State University. We looked at 2 populations. On the left is a population of over 1,000 people who we did a serosurvey in the fall of 2021. So that was during the Delta wave of COVID-19. And then we did another survey 6 months later in 2022, which was in the Omicron wave. And we measured 147 different antigens, so you can see them all lined up here. These included things like enteroviruses. It included seasonal coronaviruses, some bacteria, respiratory syncytial virus, of Epstein-Barr viruses, some of the other herpes viruses, as well as, of course, the SARS-CoV-2 virus, both the spike protein and the nucleocapsid protein. Just showing you here, if you look, we can break, these are organized by the nucleocapsid response. So here, this is the nucleocapsid level right here where I'm pointing, that you can see this group of people all had an infection with COVID-19. And then this group here were all negative for that infection. This row here is RBD, and that's largely positive because most people were vaccinated. It's the NC protein that distinguishes infection from vaccination. If you look ahead 6 months to the folks who, in the Omicron wave, you can see a lot more people by then had been infected, which probably you all remember from when Omicron came out. But this was a good test run for us. It demonstrated that we could measure responses in well over 2,000 people, all done in a single run. So you can do a lot at one time. Probably one of the most interesting findings from that study was that although there were like 1,100 people in the first collection and then 1,300 in the second collection, there were 137 people who had been participated both in the fall and the spring. So we could compare their serum from the fall to the spring and see how reliable the assay was over time. And what you can see here, looking at all of them, is that it is remarkably stable. So these are 147 different antigens, and they're virtually the same in the fall and the spring. There's almost no difference. Now, there were a few notable exceptions. I'm going to come to them in a minute. I'm just pointing them out here where things changed. And here's what those look like. So here's one example of a person, fall to spring, 147 different antibody responses and virtually no change in 6 months' time. And keep in mind that antibodies have about a 2-week half-life. So this means that the body is regulating antibody levels very precisely and keeping them very constant over time. Here's another person between the fall and the spring, and 146 of their antigens show no difference, but this one antigen changed. So this protein went up 5-fold or more than 5-fold. And we believe this person, well, we know this person had COVID. In fact, when we asked this person, between the fall and the spring, they got COVID. And that's why that antibody went up. Here's another person, fall to spring, and these 3 antibodies all went up. They're all to HPV L1 proteins, and that's because this person got vaccinated for HPV. So we can see that reflected by their change in antibody status. Here's a person who, between fall and spring, had clearly had COVID. You'll note that this person's COVID is much further down this axis than this one was. And that's because this person probably had COVID even in the fall. This person had COVID at least once. And then in the spring, this person got COVID a second time. And so, and in fact, we confirmed that by talking to the person. This person also got vaccinated for the flu, and that's why their flu antigen went up. So you can look at changes in the immune status as evidence for clinical changes in the person. Here are some observations from the people that we looked at. Couple of interesting things here. The first is looking at Epstein-Barr virus, which most people, most adults, at least in the US, have antibodies to Epstein-Barr virus, but we could see a clear progression of positivity to EBV over time. You can just look at the median value here, which is the middle bar here, and it goes up with time. The other one that was kind of interesting was rubella, which is German measles. This particular protein, the E2 glycoprotein, was much higher. This is the median for E2 glycoprotein in people over 60 compared to everybody else. And we were trying to figure out why that was. And it turns out that the vaccine for German measles came out in 1970. So all of these people were alive before 1970, and there was a pandemic of German measles in the 60s. So they probably, some of them probably were infected at that time, whereas that was not true for these other age groups. The last example I'll mention right now is just looking at the HPV responses. So this is human papillomavirus. This is the virus that causes cervical cancer. There are, as you know, vaccines now available for that. And you can see that reflected in the population data. So here you see that the 2 groups that have the strongest responses to this protein were people under 20 and people between 20 and 30. And we believe that that's primarily due to the vaccine. If you look at the gender differential, what you'll see is interesting. In the people who were 20 to 30, they're much more common to see responses in females compared to males. So here's the median for females. Here's the median for males, even though some boys were getting vaccinated. But when the recommendation changed that both males and females get vaccinated, you can see that in the under 20 crowd, there's much better balance between males and females for papilloma response. And we can also use this approach to look at, by looking at longitudinal samples, multiple samples in the same person, we can track, we can reconstruct their immune challenges. So for example, here you can see, looking at the nucleocapsid protein versus the spike protein. So nucleocapsid, you'll remember, implies infection, whereas RBD is positive both in infection and vaccination. And you can see that by looking at changes in the vectors, if you will, of these responses, people who clearly had increases in their nucleocapsid first, and then later had increases in their RBD protein, versus people who had RBD responses first, and then later moved forward to have infection. So you can sort of track what orders people happened in. And the colors here reflect exactly what we would have predicted from these longitudinal trackings based on whether they were vaccinated or infected or both. Okay, so the last topic I'm going to cover is kind of taking this to the next level. So how do we really do truly large-scale studies? And I'm going to tell you about a population study that includes over 2 million data points. So really taking immunology and immunomics to a level that it's never been at before. This is a study that we did together with several other groups as part of the SeroNet program. ASU was one of 4 nationwide centers that were building capacity for COVID-19 detection. These included Mount Sinai, ASU, the University of Minnesota, and the Feinstein group, which was a group that was a part of the health group up there. And in particular, we were interested in looking at patients who had immunosuppressed diseases. These are patients with inflammatory bowel disease, patients with autoimmune disease, patients with multiple myeloma, patients with solid organ cancers, and then patients with HIV, and then patients with solid organ transplants. And you can see we collected a variety of different samples from all these patients, totaling up to approaching 9,000 different samples from all these individuals. The samples were taken prior to vaccination and then at different time points after vaccination. Most of the samples in this particular study involve one, maybe at most two samples after the vaccination. We do have the other samples, but we haven't had a chance to test those yet. Here are some of the things we tested for. So this is a list of all the different antigens tested in this study. They include 40 different bacteria. They include 99 different species of virus, and they include 27 different autoimmune proteins. So it gives you the flavor of kind of the breadth that you can do at this kind of high throughput approach. And here's what the data look like. It's a little, overwhelming, but this is the response to 185 different proteins in these different groups. So once again, that's the healthy group: inflammatory bowel disease, autoimmune disease, multiple myeloma, solid organ malignancies, HIV, and then transplants. One thing that jumps out to you immediately is this bright box right here. You won't be surprised, though, these are antibodies to HIV proteins. So patients with HIV have antibodies to the HIV virus. You'll see there are a couple of people out here and here, maybe one here, that also have antibodies to this virus. They may be occult patients who are unaware of their HIV status. There are a couple of other things you might notice. The multiple myeloma patients sort of have a sort of dark streak running down here with kind of lower responses, as does the HIV population, and some of these other immunosuppressed populations. We think that represents people who have suppressed immune systems, and we'll come back to that in a moment. Here is kind of evidence of immunosuppression, looking at antibodies to the spike protein in SARS-CoV-2 before vaccination, after vaccination, before a booster shot, and then after a booster shot. Looking at the healthy population, you can see, generally speaking, pretty weak responses. Pre-vaccine, some of these people may have had an infection, we don't know, but clearly very strong response to vaccine in healthies that was maintained even, it maybe have drifted down a little bit pre-booster, but then kicked up nicely after the booster shot. One thing I will point out now, we'll come back to this later, is if you look in the healthy population, although the vast majority of people had very strong responses, there were some people that had pretty weak responses to the vaccine. So there may be people who believe they're healthy, but have some level of immunosuppression that prevents them from having a good response to vaccine. And of course, clinically speaking, it would be useful to identify those people, warn them, and get them additional vaccine if needed. Here you can see patients with HIV, generally speaking, had good responses, whereas people with multiple myeloma had definitely a blunted response to vaccine, although they could overcome it with the booster shot. And you can kind of look at some of these other groupings to kind of see, these are kind of the categories of kind of a weak response, a sufficient response, and a nice strong response. But definitely these syndromes, we're keeping in mind that patients who have organ transplants are often taking immunosuppressant medication, as are patients who have autoimmune disease. They're often taking immunosuppressant medication. One of the questions we wanted to ask was, do any of these antibody responses travel together? And you can imagine several different reasons why antibody responses would correlate. One would be if their sequences of their proteins were close to the same. So if there's a lot of sequence homology, an antibody to one protein will bind to another protein. Of course, if they're different proteins from the same virus and the person was infected by that virus, they're going to make antibodies to all the various proteins of that virus. And then the question is, could similar clinical states correlate with their responses to certain infections? And we think we found examples of all of those. So you can see, if you look at sequence homology, that there's this cluster right here, and we see that reflected in the antibodies. And these are all enteroviral proteins, and not surprisingly, the enteroviral proteins look a lot alike. Here's an example of infections from the same virus. These are our antibodies to different proteins in HIV, and these people who were infected with HIV are making antibodies to all of them. Here's a group of antibodies to HPV, although there is sequence homology there. This group here is kind of interesting. These are patients with cancer. They're binding to different proteins, but they are not related to each other by sequence. So presumably the clinical state of having cancer may either predispose you to certain viruses or may be caused by certain viruses. We did look for questions about whether antibodies to certain infections could predict whether someone was going to have a good immune response to the vaccine. So just to kind of carry through on that logic, the notion would be that some people may have weakened immune systems to begin with. That might be reflected in the levels of antibodies they produce against common infections. So the question was, could we detect that and perhaps use that to predict who is going to likely have a weak immune response? We generated what we call an SINR ratio, taking the top 25% spike responders and dividing that by the bottom 25% responders, so the biggest differential from top to bottom, and then look to see if they correlated with any of these infections. And as you can see, there were, out of the 180 or so proteins that we looked at, these were the ones that showed some degree of correlation. You can see that pretty much anything that showed a correlation showed it at least in the healthy population. And then it may have also showed it in some of the other populations. The darker red it is, the more likely it's highly predictive of a strong or weak response in RBD. Note that if it's red, then it's statistically significant. If we get the sort of this brown color, there's a pattern there, but it didn't reach statistical significance. We could see this especially true in the booster population, but there was some of this evidence in the other populations. And I will point out that the autoimmune and IBD population, the autoimmune population and the solid organ transplant population were kind of the outliers here. We don't know what that means, but it's possible because they're on strong medication to suppress their immune systems, that may be playing a role there. Here's kind of what the data look like in, weak and strong responders to the spike protein based on a response, in this case, to Staphylococcus aureus protein, which is a common response we found in many people, especially when you look at the booster response. Very clearly, people who have a strong response to this protein also are likely to have a strong response to the booster compared to people who have a weak response. So that allows you to do some prediction. We did see a couple of things go the other direction, which was uncommon, but it happened. In this case, not surprisingly, people who have a strong response to HIV had a weaker response to the vaccine. That kind of goes with the notion that they presumably have, AIDS or may have an AIDS-related phenomenon that weakens their immune response. Here was another response to an enterovirus protein in the HIV population that also seemed to correlate in that direction, and we're still trying to track that down. So taking all of this together, then the question we wanted to ask was, could we generate an algorithm that could predict who would likely have a weak response to a vaccine based on their antibodies prior to vaccination? So if we draw blood on them today, before they have the vaccine, can we tell which ones are going to have a strong or weak response? And The answer is we can. This is using a partially squares discriminant analysis approach. This is the ROC curve showing that we can get, an AUC around 95%. So pretty good responses here. This kind of gives you some flavor of what are the proteins that are driving this predictive model. Things like the S. aureus protein that I showed you before, respiratory syncytial virus. Some of these things have a strong pull. The grouping here in blue are the people who had strong antibody responses to the vaccine, and the grouping in red are the people who had weak responses. They had an insufficient antibody response. So we're still sorting this out, but it's kind of cool to be able to look at broad immune responses at kind of proteomic scale for the first time, and we're still learning how to do all that. So I'm going to wrap that up and just sort of thank a lot of the people who've done this work. This is Lily. She's done a lot of the work in making proteins for us. This is our lab manager for making the protein arrays. This is Deb. Ji Cho is one of our proteomics people and done a lot of the clinical proteomics in the lab. And Lu Sheng is really the mastermind behind getting the assay to work. He's the one with the golden hands. Vel Murugan helped us with all the sample collection. And Jay Park is our informatician who does all the math on this extraordinary number of samples. And then the rest of this is all members of the lab team. I'm going to stop here and stop sharing and see if there's any questions.
Thank you so much, Josh. This is a really impressive talk. Totally blew me away. So it's a throughput that we normally cannot achieve using mass spectrometry-based proteomics. So Shisha, do you have some questions to start? So you are muted.
We already have someone raise the hands. Let's get our audience questions first.
Yeah.
Xiaobo, can you?
Okay. Yeah, great.
Xiaobo raised the hand. Could you unmute yourself?
Yeah, I will allow him to talk.
Okay.
Yeah. Mm-hmm.
I know Xiaobo.
Can you hear me?
Xiaobo.
Yes,
Hey,
we're good, Xiaobo.
I guess you
Great.
cannot see me on the video.
Yeah, we
Very
cannot
nice.
see.
Very nice to see you, Josh. Yeah, and Yan Sheng. Yeah, and Shixia. Yeah, and a very interesting work. I also have a specific question. Did you calculated the correlations of the viral antibodies and the autoantibodies? That might be interesting because there are some kind of sequence homology between the viral protein and the human.
Yeah, that's a good question. We have looked a little bit at that. The plot that I showed you that correlated antigen responses did include the autoantibodies there. I don't let me see if I can pull that up again. I don't think we saw anything in this study, but keep in mind that we only looked at we only looked at 185 proteins, and only 27 of them, I think, were autoantibodies. Let me see if I can get this. Yeah. It's taking a while to. Oh, wait. Too far. There. Yeah, so the question is the human autoantibodies are on here, but I don't see them pulled out as being, correlating with viral proteins. But I think your question is a good one because it really gets to this question of mimicry. And in theory, yes, we could use it for that purpose.
Great. So there is another question from Justina. Justina, do you want to mute yourself and ask the question, or?
Yeah. Josh, thank you so much. It's really amazing the approach and the tool that you develop. So I have two questions. So as you mentioned, you have also responding the healthy. So what is your positive control for for this all those essays? Because you have to normalize something, correct? And then, okay, so please respond to this one.
No, go ahead. Go ahead. You can finish.
So the second question is, when I was looking, and you mentioned that is a really great approach for really a lot of antigens for basically doing for looking, yes, for your marker, but still ELISA, it's the golden method for this marker. So do you think so it's because of the barcoding efficiency, for example, for the low abundance molecule? Or
right. Okay. Well, so let me start with your first question. So the first question is a good one, and of course, challenging to answer. For SARS, you'll notice that a lot of our calibration was done with SARS-CoV-2, and that's because we could get samples that came from people that we knew were either infected or not infected, or vaccinated or not vaccinated. So we knew the identities, we knew the clinical case there. And especially because we have samples that predate the pandemic, and so we know they're negative. And plus there's a bunch of accepted FDA cleared assays that we could compare ourselves to. And so that's why we did a lot of comparisons there. For many of these antigens, nobody has ever developed a clinical test for them. Developing a clinical test is not trivial. It takes time, takes a lot of, getting the right samples, all that stuff. And so it's just never been done. So in a lot of cases, what we would have to do is what we call a technical positive, which is basically, taking the background control levels, going 3 standard deviations out and saying anything above that, we're going to call a positive. The other thing you can do is you can do a histogram of samples and look for, the peak that looks like the negative peak, and then look for either a shoulder or another peak that looks like a positive peak to define what's a positive. Because for a lot of these antigens, there's just no assay, and you just have to go with what you see. That said, when you see it, you'll see it. You see these responses, and it's very clear that they are quite different from the background response. And so it's the positives are convincingly positive. Where to set, the cutoff line, if it's a weak response, that becomes a little harder, but a lot of the positives are pretty obvious. I know I've already forgotten what your second question was.
It was about
the response. Right. Okay. So yeah, the strength of the ELISA responses, I think that, because we're doing in solution binding instead of, the classic ELISA is you coat the well of a dish with a protein, and the protein is sticking non-specifically to the plastic surface. So you're blocking half of the protein because it's, sticking to the plastic of the dish. Plus, you're probably denaturing the protein when you stick it to a dish. So you're going to lose conformational epitopes. We retain conformational epitopes because the proteins are naturally folded and the protein's in solution. So binding is the way it ought to be, and when antibodies bind proteins in solution. The barcode is attached via a halo tag at the end of the protein. So most of the protein should be open to the antibodies. I don't know if that answers your question.
No, it does. And I totally agree and support, floating approach for the proteins, because all of those reasons that you just mentioned. So I think so that's a great idea. And I think so a lot of people even moving into that direction. So yes, thank you so much.
Hey, Josh, I have a question.
Sure.
So this essay sounds amazing. So you could access so many of them. How have you compared with other platforms like the Olink right now seems to be very popular?
Yeah. Well, so 2 parts to that answer. So Olink is a different approach because they're not trying to measure antibodies to something. They're trying to measure the protein level itself. So it's
that's right.
it's kind of an orthogonal assay to ours. So we wouldn't compare to that. We have, however, gotten together with this group, Creative Solutions, which is part of the organization that does all the blood testing for all of the donated blood in the United States. So they look at millions of samples every year. And they wanted to do comparison studies of antibody responses. And so they took several of the big platforms like the mesoscale diagnosis platforms and some of these phage display platforms and compared this platform to those platforms. And they're putting together a paper that should be coming out, well, it hasn't been submitted yet, so it'll probably come out later this year. But the data look great. We compare very favorably with the best platforms out there, with the big difference that those platforms can study 10, maybe 20 proteins at a time, and we can do now around 1,000 at a time. So we have we can look at many more things. And because it takes just like a microliter, it's not an expensive approach.
Josh, I also curious about these longitudinal immune responses that you find out. I think that's very exciting. So have you looked into some long COVID cases? Now people are quite interested in
That'd be great. Yeah, it'd be a perfect application. Getting those samples has been a challenge for us. We really should put some pressure to get them now, because I think it would be very, very cool to look at that, including, as Xiaobo points out, autoimmune responses, to look at, the human proteins, because the viral proteins may be triggering antibodies against the human proteins. I think that would be a perfect case for that system.
So is there also is there also a application space in cancer immunity, like cancer tumor?
Yeah, we can look at autoantibodies. We haven't, we started heavily looking at the viral stuff because it was kind of easier to do.
Sure.
But we are working our way towards all of our making these proteins. They have to be in a different vector than what we use for our protein arrays, because we have to add a different tag to them. And all of our human proteins are in a different vector. And so we would have to move them all. So we're trying to figure out a way around that problem.
Yeah, makes sense.
Yeah.
Josh, I have another question. So for the assays, I think you mentioned that already it's licensed to the company. So how other people could access that? Work through the company?
Okay, so the breast cancer assay, unfortunately, right now is not available anywhere. So yeah, better to come to us for that. Certainly for this platform, we're happy to we'd love to partner with anybody who wants to do a study. So let us know and we can get together and do something for sure. We're very, very excited to do that. We also, offer the full arrays for people who want to do those. So you're also welcome to come to us for that.
That's fantastic. Thank you.
So I have a question about the cost. So the throughput is definitely amazing, right? So I guess it's difficult. It's difficult to really make it work and to have this platform available. So if you compare the cost of per sample towards like a mass spectrometry application, so what is the current cost?
So I'm going to quote a number that I got from Lu Sheng recently. I think it's something like 25 cents, US cents per protein per sample.
Per protein per sample. Okay.
Yeah. So, the more proteins you do, the more it's going to cost to get them into the library, because we have to produce a library with those proteins. And then, of course, the more samples that you do, there's a little bit of cost there too. So it's something in that ballpark. So it's, 25 cents per protein per sample is what we think the cost will be.
Okay. Did you say that you have 18,000 proteins?
We have 18,000 human proteins. We also have probably tens of thousands of different microbial proteins, including, the whole proteomes of several bacteria And then I don't remember how many viruses we have, but we have hundreds of, the entire genomes of hundreds of viruses.
Oh, okay. So the library is made. So once, oh, you still have the next gen sequence. You need to sequence that.
Well, so we have to make, for each experiment, we would have to produce the protein library for that experiment. So we would, talk to the scientists and say, which proteins do you want to include? Then we would make those proteins and barcode them, mix them together, and then we can do the experiment.
Oh, okay. So did you have already all the proteins made readily in your freezer? If that's the case, just
No, we make
take
them,
it out.
we make them
Fresh.
to make them to order, as it were. Yeah.
Oh, okay.
Because we don't want them sitting around. We want them to be fresh or reasonably fresh for the experiment.
Oh, okay. Thanks. Wow.
Hi, Josh. One information, it would be interesting to see the autoantibodies to the cytokines, just like so for the patient with HIV, these kind of patients are immunosuppressive, and there might have some kind of autoantibody to these cytokines. The activity of these autoantibodies may regulate or neutralize the activity of the cytokines, because after the viral infection, it can induce a large amount of cytokine proteins that induce inflammation. Right? That'd be interesting to see autoantibody cytokine chemokines, especially for interferon gamma.
Yeah, we do have wait, hold on a second. I lost my, I gotta get this down here for a second. We do have some cytokines in there, not a lot in this study, but let me see if I can get my zoom back here. My hold on a second. No, that's not it. Why am I not seeing this on my screen here? Hold on, I'm trying to pull up the list so you can see what cytokines I have. But for some reason, the share box is not pulling up my. Oh, here it is. Okay, I got it. Okay. Can you see the PowerPoint?
Yes.
If you look at the far right column here, we have like IL-15, IL-30, interferon alpha, interferon. So we have looked at some of the antibodies to some of these cytokines, but you're probably right. It would be good to get a much better full collection.
Yeah, to follow that request, I think so. It would be awesome, for example, to identify tumor-associated autoantibodies that will predict the checkpoint inhibitor response. Yes, for example, cancer. Yeah, because all of those, you're basically.
You see, one autoantibody is very interesting. That is cardiac myosin, right? That might be can introduce a kind of the cardiovascular disease for the autoantibodies.
Yeah, you're good. Really good point.
Yeah.
Yeah.
Great. Yeah, very interesting. Yeah, to see this detail. Yeah.
Yeah, and this great. I mean, we are talking about HUPO and humanity, and I think this is a perfect example and a great seminar. So do we have?
I have another question, just to follow up on the previous one. For Josh, for this essay, how do you address cross-reactivity? Is that a concern?
Well, what we do is we look for it, right? So, the first step is basically looking at this. And let me pull this up again. All right. So, this is how we look for cross-reactivity. And the truth is, we don't see as much of it as you would expect to see. I mean, if you look at it, the vast majority of this space was negative. The enteroviruses clearly, they had some degree of cross-reactivity. Some of these other things were really more virus related or sequence related, but and then this is kind of disease. I don't think this is cross-reactivity. I think this is just correlation. So we don't see as much as you'd think we'd see. For example, we specifically looked at the seasonal coronaviruses compared to SARS-CoV-2. And because we had longitudinal samples, we could determine when people got seasonal coronaviruses versus when they got SARS-CoV-2. And the times do not line up at all. So there's no good evidence that there's a lot of cross-reactivity there. There's a little bit with one or two of the, I guess they're both betas, but it's really not very prominent in the data.
You.
Yeah, so if there is no more question, maybe we can, yeah, thank George for this great lecture and a fantastic discussion. And thank you, everyone. Please keep an eye on our HUPO webinars coming. And thank you, George, again. This webinar is
Good
over.
to see all my friends here.
Great to see
Thank
you.
you, Josh.
Take care, everyone.
Thank you.
All right. Yeah. All right.
Thank
Bye.
you. Thank you.
Bye.
Thanks, Jensen.
Okay,
Goodbye.
bye bye.
Bye.
Okay, bye bye.